Thanks to Dr. Cameron inviting me here. Really, it has been a great opportunity for me to present my data here and also get to know a lot of people, meet some old friends also. I have been learning a lot. So today I'm going to focus on our study, mainly focus on the prevention and early detection. I will also touch a little bit of the therapeutic strategy. This is my research funding that supported me to get all the data, including R1 and 2 DoD grant I got recently with a team of people, including Dr. Tech Mark from Canada, and also, of course, Dr. Carboni, Dr. Harvey Pass, and Shreya Kanodia. Very recently, in fact, 2 days ago, I got an award letter of a new grant from EDRN. It's about a biomarker, which is what I'm mainly going to talk today. And also, I have the V Foundation grant and the Rivera Foundation grant. So as Dr. Cameron mentioned that when I started the study of mesothelioma about 15 years ago, one of the first questions is how does asbestos cause mesothelioma? So when I started, I put mesothelioma into mesothelial cells into culture. These are the primary mesothelial cells. These are totally normal cells. So when I give the cells asbestos, what I observed is cell death. Basically, after 48 hours, you see 80% cell die. So then the question is, how does asbestos cause mesothelioma if it causes majority cell deaths in vitro? So that question puzzled me and many other scientists many years. During this year's study, what we found is some critical mechanism that leads to asbestos carcinogenesis. And we published data in PNAS 2006, 2010, and also summarized with review in Clinical Cancer Research 2011. Basically, summarize what we found is in vitro chrysolite, which is the most carcinogenic type of asbestos, kills primary mesothelial cells. The cell die, and it dies through a process is necrotic cell death. And that type of cell death basically release a protein called HMGB1. That protein is critical. It's like a switch on of the immune response. It causes inflammatory cells, including macrophages especially, accumulate in that area. And those immune cells secrete cytokine, especially TNF-alpha. The cytokine binds to receptor and activate kappa B and some other cell survival signaling pathway. So the cell no longer dying, and instead they survive, but survive carry the damage caused by asbestos. In the long run, you have a pool of basically damaged surviving cells, and they can be transformed in the end. So basically, this pinpoint HMGB1 is really a critical factor initiate all this process. And also immune cells, and especially some cytokine TNF-alpha plays a critical role. So this shows another experiment we did. As you see, if just give the cell asbestos, the cell die. However, you put the TNF-alpha cytokine together with the fiber, the cell can survive. This is after 1 week, you see still a lot of cells alive. And after 1 month, you start see this kind of foci forming, suggesting the cells getting transformed. We further did experiment to test the importance of HMG1, that molecule. And here we use co-culture. We put macrophages on the insert chamber and put primary mesothelial cells, normal cells, in the bottom. And this insert have those pores can allow these two cells interact and exchange molecules. And we put asbestos in the system. And you can see that after this take about 1.5 months, we start seeing foci formation in the mesothelial cells. As I mentioned, HMGB1 we think is critical molecule. So we try to target and inhibit HMGB1 function. Using here, we use BOX-A, which is antagonist, or use anti-HMGB1 antibody, which also inhibit the function. You can see by using those inhibitors of HMGB1, we can inhibit foci formation. So HMG1 is a very interesting protein. In fact, when it was discovered, it was found in nucleus, and there is a huge amount of HMG1 in the nucleus. For a while, people call it even housekeeping gene because besides histone, this is the largest amount in the nucleus. And more recently, in fact, started 1998, people found that HMG1 not only stays in the nucleus, it can be released. First, they found depends on cell death, necrotic cell death, release HMG1 out of the cells, and it become pro-inflammatory factor. However, apoptosis normally don't release it. That is why apoptosis, you seldom see inflammation. And also then they found that some immune cells can secrete, and especially some tumor cells, what we discovered, can also activate secrete HMGB1. That I'm going to show you the data later. So as I mentioned that asbestos kills mesothelial cells, and in the killing process, HMG1 is released. Here is one of the immunofluorescent result. You can see untreated cells, the normal culture cells, HMG1 stay in the nucleus. If you induce a cell with apoptotic inducer, which here we use actinomycin D, although the cell are dying, HMG1 still in the nucleus. Instead, if you give the cell asbestos, the cell die of necrosis, HMG1 got released, and in fact go to cytosol and then further go out of the cells. Here is an animal experiment. We basically inject asbestos into peritoneal cavity of the mice, and after 2 months, we got the tissue and did staining. You can see there is a huge amount of chronic inflammation in the area where there is asbestos deposits. And if you stain those chronic inflammation area, you can see the staining of HMGB1 and also TNF-alpha is very high. And we further checked the serum to see whether HMGB1 level also goes high in the blood. And we inject also different group of mice. Here we use glass bead as an active control. Then we use different type of fibers, including chrysolite, crocellite, and different type of arenite. These are all carcinogenic fiber. You can see they all induce HMG1 in the blood. Then the next question is, how about human being? Since we see such a good result in the animal model. So luckily, with collaboration with Dr. Harvey Paz at NYU, he provided me the serum samples from different populations. We got unexposed healthy people serum, and also heavy smokers who had lung inflammation, together with the people who has been exposed to asbestos. Those are the workers who has occupational exposure to asbestos. So we checked HMGB1 in their serum. We found that those people who has been exposed to asbestos have elevated levels of HMGB1 in the blood. So this suggests that HMG1 can be probably a biomarker of asbestos exposure. Then we further test how about tumor, how about mesothelioma tumor specimen. So here on the left is normal pleural. These are normal tissue. You can see HMG1 only stays in the nucleus and the level is low. However, if you compare to the tumor tissue, HMG1 level in the tumor is extremely high, not only in the nucleus but also in the extracellular space. So we did a bunch of experiments in the lab. First, we also tested mesothelioma tumor cell lines compared with primary cultured normal cells. And we found the mesothelioma cell lines express high level of HMG1. And HMG1 not only in the nucleus, but also in the cytosol, instead of normal cell, mainly in the nucleus. And we also tested the tumor cells, the cell culture medium. We found that tumor cells can secrete actively HMG1 to extracellular space. So those cells are not dying, they are alive, but somehow they can actively secreting HMG1 to extracellular space. That's suggesting the cells need that in the microenvironment in order to grow. And we did a bunch of experiments. Here I don't have time to show everything. In summary, basically we found that HMG1 is critical molecule for the tumor to grow. And targeting HMG1 can be a therapeutic strategy. Here is one of the experiments we did. We did a Malignant assay. So basically you made a scratch. After 48 hours, you see the scratch heal. However, if you target using HMGB1 inhibitor, including BOX-A, which is an antagonist, or HMG1 antibody, or RAGE, which is a receptor of HMG1, or the RAGE antibody, all these targeting HMG1 function, you can see it can inhibit wound healing of the cells. Here is a soft agar assay. Similar, if you don't do anything, you see the cell form a ball basically in the soft agar, form a colony. However, if you target HMG1, no matter you use BOX-A or the antibody against HMG1 or antibody against the receptor, you see the number of colony or the size all got reduced. We further did animal. This is xenograft. Basically, we inject human mesothelioma cells to skin mice, and the cells express luciferase. So using IVIS machine, we are able to monitor the tumor growth. And you see the PBS, basically no treatment control, the tumor grows very fast. However, if you target HMG1, using here we use box A, which is HMG1 antagonist, the tumor growth is inhibited. And this is survival curve, again, compared to untreated PBS. The box A treated mice have much better survival. And in fact, one of the mice in the end, we couldn't detect even any tumor. That mouse survived 2 years almost. So basically, we published the data in Cancer Research, and we are lucky were chosen as the front page of that issue in 2012. So as I said, that we found that HMG1 is critical molecule, and chronic inflammation is in fact very important in asbestos carcinogenesis. So therefore, if we can find some anti-inflammatory drug can target the inflammation induced by asbestos, we might be able to prevent and treat mesothelioma. So among all the anti-inflammatory drugs, one of the first one we thought test is aspirin. As you know that aspirin has very low toxicity, and it has been proven have the efficacy in inducing many cancer, including colorectal and liver cancer, even breast cancer. And there is a famous study called a Physician Health Study, and basically it's aspirin trial. In that study, about 22,000 physicians were followed for 24 years. They were even gave placebo or 325 aspirin on an alternative day. So with the help of research in Harvard, we got access of this data. So I asked the collaborator, I said, can you help me to analyze in that population whether there is mesothelioma patient and whether you can see any difference between aspirin taking group versus placebo. And then she told me the result. She said, among them, total 70 cases. Unfortunately, the number is low because mesothelioma is a very rare cancer. So there are 17 cases, and in comparing aspirin taking group versus placebo, she did find 30% reduction of the risk, which is good. However, unfortunately, because the number is so small, there is no enough significant power to show the significant difference. However, the trend is there. So that is very encouraging. So we further did an experiment in the lab to test whether aspirin can inhibit HMGB1 or reduce HMGB1 level in the mice. Here you can see that, as I showed before, once you inject fiber to the mice, HMGB1 level goes high. However, if you give aspirin to the mice, it significantly induces HMGB1 level in the blood. This is another animal experiment we did. We inject tumor cells into the mice, and with the tumor growth, HMG1 also goes high. However, if you give aspirin to those mice, you also see the reduction of HMG1 in the serum of the mice. This bunch of molecular biology, cell biology experiment, basically we show that aspirin and its metabolite, salicylic acid, can inhibit HMG1 dependent growth of mesothelioma. Here, basically, we use a couple of mesothelioma lines. We compared aspirin treatment versus PBS, the progression and the effect of progression, migration, and invasion. We found that by giving the cell aspirin or salicylic acid, we can induce cell growth, migration, and invasion of the tumor. This is an animal model we did using mice. Again, this is a xenograft model. We inject mesothelioma cells into the skin mice, and with IVIS machine, we can monitor the tumor growth. So we gave the mice either control trial or you give aspirin with 2 doses. One is 25 milligram per kilo or 50 milligram per kilo. So according to the literature of previous study, 25 milligram per kilo in the mice is equal to baby aspirin to the human. So you can see that either low dose or high dose, both can inhibit tumor growth and increase survival. So this result is very encouraging. So basically suggests aspirin have this effect of anti-HMG1 also probably can be a good prevention or even the early treatment therapeutic strategy. So in the next part, I'm going to touch a little bit about another topic that led by Dr. Carboni in my group. So basically, me and Dr. Carboni have been working a lot on the genetic factor in mesothelioma, and Dr. Carboni is a pioneer in that study. What happened is basically, besides those spontaneous mesothelioma, we also noticed there are some familiar mesothelioma cases, especially in some area in Cappadocia, Turkey, and also there are some American families. We mainly started focus on W and L family in the United States. So these are the pedigree of these two families. All this icon basically symbol is mesothelioma patient. You can see both families have a lot of mesothelioma patients. Considering mesothelioma is such a rare cancer, when you see so many patients in one family, that clearly suggests there's probably something else besides asbestos exposure. So during years of study, again, in collaboration with Dr. Testa in Fox Chase, we discovered that germline BOP1 mutations predispose to mesothelioma. Basically, this says that once the BOP1 gene is mutated in those people, the function of BOP1 got interrupted. Basically, the protein will no longer go to nucleus, and basically, the people are easy to get mesothelioma if they are exposed to asbestos. And we found that this is not only limited to mesothelioma. BOP1 mutation can cause a cancer syndrome. So including various cancer, including mesothelioma, uveal melanoma, cutaneous melanoma, and also MAP-based, which is a skin lesion, together with renal cancer, breast cancer, and probably some other cancer we saw in those families. So it's a big cancer syndrome that now more and more people are very interested in this type of study. And we did also some animal experiment testing the function of BOP1 predispose the individual to mesothelioma development. What we use is the mice have heterozygous BOP1 deletion. And you can see in those mice compared to wild type, if we give normal dose of asbestos inject into the mice, in the wild type mice, you see certain like 20-something% of mesothelioma development, but in those BOP1 mutant mice, you see much higher incidence. And if we reduce the dose 10 times less, which is 0.5 milligram of asbestos in wild type mice, we seldom see tumor. However, in those mutant mice with no BOP1, with less BOP1, you can see the incidence is very high. And this suggests that BOP1 mutation can definitely predispose people to asbestos development. And then we further did more study, and also with more collaboration, more interaction with people from the scientists from different fields, we built a big basically pedigree and cohorts of a lot of BOP1 mutant carriers. And more recently, we published in PLOS Genetics, basically we combined genetic and genealogy studies. We uncover a large BOP1 cancer syndrome kindred, tracing back 9 generations to a common ancestor from the 1700s. So basically, this is a huge pedigree. I only choose part of it. In fact, there are a lot of branches. So basically, hundreds of people we connected. And this, in fact, started from these 4 individuals. These 4 individuals are referred to us by different doctors. And they carry, later we found that they carry the exactly identical BOP1 mutation. So normally we saw different mutation on BOP1 side, that gene. However, these 4 individuals carry the exactly same mutation. So in the beginning when we saw that, we said, okay, there are 2 possibilities. One is that is a very hot spot, got hit easily, got mutated. All these 4 individuals are somehow related, although they don't know because they even live in different states. So by working together with a lot of doctors and scientists, we finally really linked these 4 families to a common ancestor. So this is exciting, but in the meantime, we also catched a lot of people carrying BOP1 mutation who they didn't know before. And this is an issue because they know that they have the higher risk to develop cancer. Is there anything we can do? So as we know that early detection normally leads to better survival. So if we have some tools to detect disease at the early stage, we might be able to save life. So as I showed previously, we found HMGB1 is critical protein and it gets elevated in the individual who exposed to asbestos. We further tested mesothelioma patient compared to normal people. We found that HMGB1 serum level in mesothelioma patient also goes high. And we also compared serum and plasma versus normal people. We also noticed that HMGB1 always go high in the mesothelioma patients. So now we are conducting a screening program, basically routine check those people have higher risk, especially those BAP1 mutant carrier. Hope that HMGB1 can be a good marker for even early detection. So this is still ongoing. I hope we have some nice data to present next time. Again, talking about HMGB1 is a very critical protein, and also it's a very interesting protein because not only the localization comes to different function, but also recently, in fact, in the recent 5 years, new discovery found that HMG1 have also different isoform. So there are, depends on acetylation, there are different acetylation isoform, or depends on redox status, is also different redox-related isoform. Today I will mainly talk about acetylation status-related isoform. So what people found that if the HMG1 is actively secreted, it is hyperacetylated in the nuclear localization site because HMG1 normally stay in the nucleus. However, there is transport from nucleus to the cytoplasm. So you need acetylation to keep the cell in the cytoplasm, otherwise it will be reversed. So once it's acylated, it stays in the cytosol, then later get secreted outside of the cells. If it is passive release, basically the cell die and release nuclear HMG1 to extracellular space, those nuclear HMG1 is non-acylated isoform. So basically, by comparing by recognizing whether it's acylated, hyperacylated, or non-acylated, you know whether it's passive release or active secretion. So with that information, I start thinking about our mesothelioma model. As I showed previously, when primary cell die because of asbestos exposure, it is necrosis, it's passively released. So I think that at that time I thought, that probably is non-acylated HMGB1. However, in mesothelioma patient, because those are the tumor cell actively secreting HMGB1, and that actively secreted HMGB1 should be hyperacetylated. So is that right or wrong? So we did analysis. Luckily, I got a calibration from Dr. Dan Antonio from Liverpool. He's expert of mass spec. So using mass spec, he's able to detect different isoform of HMGB1. So exactly like what we expected. So when you give primary cell to asbestos, the cell die passively, and we detected non-acylated HMGB1. However, in normal culture, the tumor cells, we see a lot of hyperacylated HMG1 in the cell culture medium. So with that, we start thinking, how about a patient? Or compared to asbestos-exposed high-risk cohorts, can we see difference of different isoform? And again, thanks to Dr. Harvey Pass gave me the samples. We did a collaboration. We measured HMG1 using mass spec, including total HMG1 and also hyperacetylated HMG1. Well, you can see that total HMG1 goes up in the people exposed to asbestos, although they don't have mesothelioma yet, but you already see HMG1 goes up. And in mesothelioma tumor patient, you see further up of total HMG1 level. If you look at a hyperacetylated HMG1, that isoform, the mesothelioma tumor patient has significantly higher. And in fact, the cutoff is about 2. With that cutoff, we could even have basically 100% sensitivity and 100% specificity in that population basically we tested. Although the number is relatively small, however, the result is very encouraging. That is why we want to conduct a large clinical trial, hopefully validate the result. Also, we found that compare early stage to late stage, we didn't see big difference, suggesting that probably it can be also a good tool for early detection because we could see relatively good amount of HMG1 even at an early stage tumor. So to summarize, what we found is when people are exposed to asbestos, the cell die passively, release an unacetylated HMG1 into the extracellular space we can detect in the blood. However, if there is tumor growing inside the body, the tumor cell secrete hyperacetylated HMG1. So by detecting that, we can detect mesothelioma. So this result was summarized in Clinical Cancer Research this year. And we also tested other biomarkers, including mesothelin, fibrillin, and osteopontin. Those biomarkers are previously proposed by different researchers, and some of them got very exciting result also. So we were thinking by combining HMG1 and fibrillin, then we combine HMG1 with different biomarker. We found that with fibrillin, we got the best result. And this experiment, in fact, showed we tested that in different patient cohorts, including mesothelioma, all patients with benign pleural effusion, all patients with malignant pleural effusion, but not mesothelioma. So we have these three different groups test HMGB1, either total HMG1 or acylated HMGB1. We notice that by combining with fibrillin 3, we got the best sensitivity and specificity of this detection, suggesting that probably this combination will be even better biomarker in the future clinical trial. As I mentioned, that there are a lot of questions still need to be addressed. First, we need to use a bigger cohort to validate the result, and also we would like to understand better of the function of HMG1, especially different isoform of HMG1, to see what its contribution to the asbestos carcinogenesis and mesothelioma development. So luckily with a team of people, especially my mentor, Dr. Michele Garbone, and also Dr. Tech Mark, a very distinguished researcher from Toronto, and Dr. Harvey Pass at NYU, and Dr. Canadio at Cedars-Sinai. We formed a team and we applied the DoD Team Award, and very, very fortunate we got it. So in that award, we are going to determine whether asbestos pathogenesis require HMG1, also require what type of isoform HMG1. So we are going to, TechMark is going to provide all different type of mouse model generating different isoform HMG1, allow us to answer those questions. And also we want to further to, as I mentioned, to test HMG1 in the larger cohorts, especially we are going to do a retrospective trial and also a prospective trial to test this as a biomarker. Again, this is truly teamwork. This is our lab in Hawaii, beautiful place. This in fact is in the backyard of Dr. Garbone. So I'm very lucky to have all those great people working together. Thank you very much.